Okay, we're going to start out today using uh, talking about the Golgi complex or Golgi apparatus. The Golgi complex or apparatus is named because it's a series of membrane structures, they call it pancakes, membrane stacks, where a lot of different activity occurs. There is an ordering of the stacks from the nearest, which is called the cis, all the way through the medial to the trans, which is furthest away. And so the journey for biosynthetically made proteins starts in the certainly in the ER, and you have the ribosomes, and they deliver the protein inside the ER. Vesicles then take that protein that's been made and fuse with the cis, and they call it a cis network. It's kind of a loose tubular vesicle network, and it's thought to be the end of the ER, the beginning of the Golgi. It's kind of hard to determine there. But anyway, the protein is delivered to the cis pancakes, cis stacks. They then are moved either through maturation or vesicular transport or both into the medial and then to the trans, and there's different fates that await certain proteins from the trans Golgi network here. Get my pen going here. Okay, we're all happy. Yes. Okay, so let me stop and see if this recorded. Okay, looks like we're continuing with our Golgi structural look. This cartoon here, of course it's kind of backwards here from this. So the ER would be here. ER is here. And there's the cis face, and that's known as the cis Golgi network, to the cisternae, and then the trans Golgi network. Cisternae are stacks that can actually be purified. If you remember the original cell-free system, these stacks are actually what Bosch and his group did, is they purified these stacks from the mutant and the wild type cell lines. And so you have vesicles that are moving from the ER into the cis Golgi network, into the cis cisternae, medial and trans, and then out through the trans Golgi network. Okay, and you get different fates these vesicles can undergo. This is when the structure of the Golgi next to the ER, that's the stack concept. And you can see they look like a bunch of stacks. If you do some immunofluorescence, here's material inside the Golgi stacks, and they're showing it near the nucleus, near the ERs, right around the nucleus. Its Golgis are located near the cell nucleus, flattened sacs called cisternae, and they have a cis face, which is also known as an entry face, and then a trans face, which is an exit face, and vesicles that will be leaving the Golgi and moving and carrying cargo to other destinations. So the function of the Golgi is to transport and process secreted cytosolic proteins, enzymes, hormones, transmitters, these kind of things, and membrane proteins newly made from the ER to the cell surface. So this cartoon shows the Golgi cisternae, the cis medial trans. Here's the trans, the cis medial network. They're not showing it here. Cis Golgi Golgi network, CGN, and this is the trans-Golgi network. And so vesicles certainly are made on ribosomes that become attached, and protein goes in to the ER and then is packaged and delivered to the Golgi. And if you remember, we talked a little bit about carbohydrates and the high mannose Christmas tree that were decorated on asparagine X serine or asparagine X threone areas. And we talked a little bit about how that process works. There's a dialcohol molecule that actually enzymes that take from the built tree and stick it onto the protein at an asparagine. So these are known as asparagine, and asparagine has the one-letter code N. So it's N oligosaccharide linked. Okay, so they'll call it N for asparagine linked carbohydrates. Polysaccharides mean multiple sugars, so there's multiple sugars in the tree. It's a simple tree because it's pretty much some mannose and glucose. There's initial trimming that goes on in the ER, but that trimming then goes further. And if we're talking about lysosomal enzymes, there's some phosphorylation that occurs in the cis Golgi that actually tag lysosomal enzymes. Their carbohydrates end up with a mannose 6-phosphate residue that's a target in zip code signal to go to the lysosome. Other proteins have carbohydrates that you get removal of the mannose in addition of more sophisticated sugars, a little bit more complex. The gluten X sugars, the galactose sugars, and the NANA are known as silylic acid is NANA. Okay, neuraminidase also has silylic acid here. And so you end up getting a transport of glycoproteins through, and you can actually track the protein. If you purify the stacks, you can actually track a protein to see how long it takes to get fully glycosylated from the simple high mannose form to the complex, which has gluten X, galactose, and NANA sugar residues. So I suspect in our personalized medicine, they'll be able to figure out how efficient our Golgis are, and that might give us information into potential for diseases. If your Golgi is less efficient than others, you have bad Golgi enzymes, as it were. I figure that's coming. And there are certain fates that happen. 3 big fates as you end up going to maybe the lysosomal system through the endosomes. You may end up being a secretory granule, and that's regulated secretion, or you may just, some material may just move out and the vesicles fuse with plasma and dump material out the cell. So you have kind of the biosynthetic versus the secretory. So it's 2 different journeys. The biosynthetic is that you're making materials that end up in some place in the cell. And to go a little further, how does a Golgi protein know to stay in the Golgi and not get on and hitch a ride? Okay, so there's certainly anchoring signals and domains on ER proteins. Golgi has anchoring information. There's zip codes to endosomes, lysosomes. And then secretion pathway is vesicles are with cargo that is going to be released, either released right away, that's constitutive, or regulated, requires a signal to fuse with the plasma membrane and release, say, in nerves, neurotransmitters. So all our nerves do regulated secretion. So biosynthetic is movement of material from one of the organelles and either stopping or moving eventually to the lysosome if it goes all the way here, if it enters the biosynthetic pathway, or regulated or constitutive secretion. And so what's interesting is, how do proteins get into the right trains, right? If this is a train station, how do you catch the right vesicle to go where you need to go? So there's a lot of information on sorting. See that word sorting? It's easy to think about, but a little bit more complex to understand how that actually occurs. And they're still researching on it. So here is shown the little GIF down below. So here's the vesicle. Here it is, fused and releasing cargo here at this point. There it is. Yay. So again, there's the biosynthetic pathway. Biosynthetic. Okay. I know, BS. Dr. Bush BSing us here with the biosynthetic pathway. But there is secretion that occurs, and it's unregulated, means the vesicles just quickly fuse, and that's called constitutive, which means always on or always active. So you get a little bit of material moving out. Notice that there are membrane proteins being carried out, lipids being carried out. So if you think of material coming in, that material has to get replaced, right? The membranes, I mean, if you didn't replace it, the cell would get smaller and smaller. So this is a lipid pathway as well, that you replenish the lipids on the plasma membrane, and you get various receptors and things sitting on the surface. Constitutive. Regulated means the cargo is separated and ends up in secretory granules. And the reason they call them granules, when they do the histology, they're so electron dense that they appear just dark in a transmission electron. In other words, there's probably very little space or material in the granule for anything else. These proteins are stacked, probably almost crystallized in how concentrated they are. And then in the presence of a signal such as a hormone or neurotransmitter, there's a cascade of protein signals, kinases, phosphatases, that then triggers the machinery to fuse. And so right now, this little cartoon, there's a bunch of proteins and materials required they're not showing. Okay, but part of this, what I study, I actually study Rab proteins, which are involved in this process. So this is what I've been studying most of my life, is Rab proteins, RAB, rat brain associated. It's where they found it first Rabs. So Rab, rat brain associated. If you ever want a rat associated brain or whatever, they decided to call it a Rab. It's a cousin of RAS, Rho, Rick, ran bunch of different GTPases. So the biosynthetic pathway is connects with the endocytic system. Endo means internalized, so material is taken into the cell, and the material can end up in the lysosomes, as well as in the biosynthetic pathway coming from the made in the ER to the Golgi and to the lysosome. Enzymes are there. So the lysosome has a bunch of different functions, but it's very acidic and has a lot of enzymes. And in our white blood cells, these lysosomes will break apart, invading bacteria here. And so here is showing the biosynthetic pathway, the BS pathway. Yeah, BS. Boy, I can't spell. And then here's one of the secretion pathway. This is constitutive here. But this is the biosynthetic and unit vesicles that can fuse with early, or in this case, late endosomes, and the vesicles can move to lysosomes, or material comes in through endocytosis, and there is some evidence that there's a maturation. What that means is lipids and proteins are taken away and added to turn the early endosome into a late endosome into a lysosome. So there's this maturation model out there that the cell actually takes early endosomes, puts them together with vesicles coming in, and then remodels them to a late. So there are protein markers that are only found in certain organelles. And early endosomes, late endosomes, and lysosomes have different markers. They have material in common, but they have material that is different as well. And they're not shown, but there's also movement back out that's called exocytosis. Exocytosis. So they can actually track a receptor, and they can actually track how long it takes to release its, say, its transmitter or whatnot, and be spit back out. So velocity of all this will eventually be measured by the bioinformatics, and we'll see how good your receptors flow or how good your signal transduction is. This is all coming. This is going to be information. Maybe, you guys, girls going to medical school, that you'll actually have a patient, and that may change how you treat them, is depending on how good their cells work. And I suspect they'll probably do this over in aging studies as well. Is that part? I would predict it's part of aging. So they'll have aging informatics. What happens to your cells when you were young and your Golgi was working well, then it slows down. Is that part of a disease process here? Eventually, lysosomes can be the dead end for the biosynthetic pathway. They will recycle materials. You can have materials broken up in the lysosomes. When materials such as mitochondria are chewed up, it's called autoselfphagy, which is phagocytosis. Okay, and here's the membrane actually wrapped by parts of the ER, wrapped around a damaged membrane, and that confused with lysosomes, and the lysosomes break down all the enzymes and material, and you recycle all the monomers back for more enzymes, lipids, sugars, etc. White blood cells take bacteria in, wrap a phagosome, and then you have a fusion, and you hopefully break down the bacteria. And material can come in through the early, the late, and lysosomes. So you can have material being moved in and potentially destroyed as well. And again, they're not showing exocytosis here, but that can occur as well. Lysosomes are very acidic, pH 5 or less. Here's pH 5. Cytosol's about pH 7.2. And they have a bunch of enzymes that chew up all sorts of things. Notice ACEs, nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases, all sorts of enzymes that would chew up lipids, nucleic acids, sugars, proteins. The hydrogens are pumped in by an enzyme known as an ATPase. It's kind of a reverse of what happens in the electron transport system, where ATPA is, in fact, ATP is broken down, and that energy drives the movement of hydrogen ions in to make it acidic. And so you end up getting a collection of hydrolytic enzymes in a very acidic environment. So that's the environment these work at the best. So the pH optimum for these enzymes tends to be very acidic. Okay, so we're taking the first look at our cytoskeleton, just a brief overview. And the cytoskeleton has 3 different flavors of cytoskeleton. This determines the shape, the mechanical nature of our cells. Can they undergo stress or movement, motility? Think about mitosis and movement of chromosomes and other materials from the mother cell to the daughter cell and mitosis. All that's through the cytoskeleton. Pathologists look at cell cytoskeleton to determine is it cancerous or not. Cancer cells tend to have an abnormal cytoskeleton. They look different. So a pathologist will do a biopsy and look for things like abnormal cytoskeleton. Whoa, will that go back? Well, the 3 components of the cytoskeleton are actin, microtubules, and intermediate filaments. These hold the cell's volume, allow it to stretch, to move, and do motility. And I forgot what else it did. But anyway, you can read the slide before. This is an immunofluorescence showing how we have fluorescent markers. And we can see that the structure of these intermediate filaments, microtubules, and actin are completely different from each other and localized in various specific ways. And certain wild type cells and cancer cells would have different looking structures here. Common features, they are polymers. Usually there's a monomer that are put together. So there's actin, intermediate filaments, and microtubules from small to very large in terms of size. Our cytoskeleton is dynamic. Each one of these can assemble and disassemble and are very highly conserved in structure, which is good. We want all our cells to behave the same. So let's talk intermediate filaments. Here's intermediate filaments. How do we know what they look like? We can actually visualize here. And it looks like a twisted rope, a bunch of Twizzlers kind of coming together. And they span the entire cytoplasm and are actually anchored in specific spots between a monolayer of cells. And the anchoring allows cells to stretch. Here's shown as stretching here, and the cells stay intact. And if we didn't have intermediate filaments, cells would rupture. Okay, so they withstand mechanical stress, intermediate filaments. So you have a long alpha helical region of the filament protein. It coils into a dimer, and that's a coiled coil. And then staggered tetramers form together, and 2 of those come together as 8, and 8 tetramers will be twisted into this rope concept here, and that is a filament. So this can be unwound, and this can be synthesized. So the cell obviously is modulating and changing the filaments based on proteins and materials that regulate the winding and the coiling and the uncoiling of the filaments here. The next type of cytoskeleton is the microtubule cytoskeleton. It gives us cell volume. Actin fibers are concentrated at the periphery. Most microtubules radiate from a central location in the cell, and the main function is transport and very required for mitosis. And it shows the centrosome complex that you're aware of in mitosis. They provide an organizing structure in interphase cells, and in mitosis they're in blue are the chromosomes lining up here in the metaphase, and then anaphase, you're breaking apart the sister chromatids, and there's a pulling mechanism. The filters are actually then contracting and moving the material through the cytoplasm until you end up getting cytokinesis and breaking apart the two daughter cells. Microtubules also provide tracks for vesicle transport, especially in regulated secretion. These red vacuoles contain regulated neurotransmitter granules, and they sit at the axon and jump the synapse only when there is a signal, and that then turns on the machinery to fuse, and the neurotransmitters, okay, jump the junction, and there's a receptor, okay, on the other side that picks them up and starts the process of moving another signal down to another neuron. There's also vesicles that can go in the opposite direction, and remember, the plus is where the tubules are added to, the minus are where they can be shortened or lengthened, and there are vesicles that move down, and there are vesicles that move up these kind of highways. So you have inward transport or outward transport, and the microtubules provide these highways here. And obviously there's the molecular motors and machinery that are involved in moving these vesicles to and from in neurons here. They're made of tubulins, that's why microtubules. Tubulin is the protein. There's three flavors. Two are the alpha and the beta that form a dimer, and these dimers come together to form a protofilament, and 13 of the protofilaments wrapped around like a cylinder, and there's a hollow end, and that's the wrapping, and the building is at the plus end, and these filaments are broken down at the minus end, and they form the alpha and beta tubulin, and they represent the building blocks. So you can add to it, to the plus end, or take away from the minus end. How you stop the growing: there is a specialized protein that will add a GTP to the end of the plus side, and that stops the growing and stabilizes the ends, so there's no the GTP will be on either and cap them. And if you want to start growing or changing the structure, you have enzymes and machinery that remove the GTP cap. Okay, so there's other machinery that's not showed here in the microtubules. Here are the motor proteins, bind to microtubules, and they use energy. This is the plus, so this is the axon ends from the plus to the minus nearest the nucleus here, and you have dynein moving vesicles in this direction inward, and you have kinesin moving vesicles outward here, and the vesicles are attached to these protein structures, and you end up getting movement down these microtubules. So there's clearly proteins that interact. So if we were doing protein interaction processes, we would actually could find that these proteins on these motors will interact with the microtubule proteins as well. And that changes the conformation based on energy. So hydrolyzation of the ATP to ADP, and that causes the conformation change, and these kind of to walk down the microtubules. I think there's some movies out there you might want to check out. Pretty neat. Finally, the last is the actin that interacts with the myosin, and the actin is the most common protein in all the cytoplasm. So if you do a protein purification, you break tissues, cells up, actin should be as a control. If you can't get a lot of actin, something's wrong. They're concentrated but on the edges, and they give the cell mechanical strength. They determine the cell size and cell movement. White blood cells and whatnot need the actin changing. Cancer cells will have abnormal actin, and they will look different in a biopsy. So a pathologist can just look at the size or the shape, say, well, this is something abnormal here. And they can form with another protein, myosin, and twist around, and you end up getting a contraction, kind of like a mini cellular muscle, and these cells can move. So with white blood cells, the movement actually can attract them to an infection area. They follow regulators of inflammation and try to wind up at a scene where bacteria or viruses are causing a ruckus in the cell. So there's a specialized kind of fourth type of cytoskeleton, but it's only in red blood cells, and it's a specialized function to make the red blood cells concave, look like little donuts. And it's the spectrin is an alpha and beta chain that's twisted, and it attaches to the inner leaflet, and that allows the red blood cells to go into the smallest capillaries and veins and move through and deliver the oxygen or pick up then the carbon dioxide and undergo the physiology of, allows us to breathe. And if a red blood cell doesn't have spectrin, it's not as efficient. So again, how efficient is your spectrin? Maybe people will tend to have more severe breathing disorders if their spectrin is not as good as others. So we could predict that. They may have to be treated differently. That's all coming. How do they know what it looks like? They've actually visualized the spectrin here, and in an electron microscope. And the spectrin binds to specialized areas, and they're showing these actin molecules are there, the spectrin dimers, and these things twist and connect these anchored sites called a junctional complex, bunch of proteins here that allow them to sit underneath and hold the red blood cells into their proper shape and conformation there. So we'll in lecture 3 we'll talk about then the lipid side, and phospholipids are not just random lipids floating around. There are protein and cholesterol areas that are known as lipid rafts, and we'll talk to you all about that. So come back soon and check out the membrane lecture 3.